Hi, everyone. Welcome back in. We're going to start the next session. It's my pleasure to introduce Ali Mortazavi, who's here from UCI.
All right. Thank you for the introduction. So I'm going to tell you about a developing story, which is our efforts to analyze the long-read transcriptome data from ENCODE4. And I realized that there's a lot of you in this room who are part of ENCODE, and so it shouldn't need an introduction, but at the same time, I thought it's worthwhile, now that the project is officially over, to sort of see what's happened over the last 19 years, and it'll probably be 20 years by the time the last set of papers come out, and to see how this one project has actually driven quite a bit of the genomics infrastructure that we use for day-to-day functional genomics. So the original pilot came out of actually the end of the human genome sequencing project, where we had the sequence of a genome and very little idea what it was all about, right? And so the idea was that we had a puzzle. We have the 3 billion pieces of it. And the question was, where were the significant portions of it? And at the time, we did not have the platforms to be able to actually do whole genome analysis. This was the time when you could do whole genome maybe in a yeast, maybe in a fly, maybe. But realistically, when you were talking about doing analysis in human, the whole pilot phase was based on trying to do the analysis of 1% of the human genome, right? With the thought that we could pick a set of different regions, learn something about them using a variety of techniques, and hopefully figure out some way to then take this project to the next phase, which was always going to be to scale up to the whole genome. And the one set of techniques that really did enable ENCODE to scale genome-wide was the development of next-generation sequencing and of techniques such as ChIP-seq and RNA-seq, which were developed really as part of the ramp-up for ENCODE2. So I remember those days pretty well. A lot of the different groups that were proposing projects for ENCODE2 were still talking about doing macroarrays. One group proposed to do ChIP-seq and RNA-seq. By the time they revised all their proposals, when it came time to funding, they had all switched to doing ChIP-seq and RNA-seq. This thing caught up really quickly. And ever since then, we've been doing this. Now, ENCODE2 famously was cell line based. And I think in the mind of a lot of the genomics community, it's still primarily about cell lines, right? Hundreds of ChIP-seq data sets in K562 and HepG2, for example. But ENCODE3 decided to take this encyclopedia term that was part of the clever naming that at least Feingold had come up with, and try to actually concretize it, make it a concrete actual encyclopedia of regulatory element. And so people such as Xiping, who's in the audience, took the lead to actually build an encyclopedia of candidate regulatory elements. Now, we also at that point started adding data from human tissues. There was also a really beautiful embryonic mouse time course that came here. And those papers describing the effort that finished in 2017 just were published in 2020. And so that's probably what most of you have heard about. Now, the project did go through a fourth and final phase. And I want to talk a little bit about one part of it, which is the part I'm involved with, which is adding long reads to the RNA-seq part of ENCODE. But I would be remiss if I didn't mention that in addition to the main ENCODE efforts, a whole set of other projects came out of it. For example, GENCODE started actually as a part of ENCODE and then became its own project, as well as, for example, the lrRNA-seq Consortium, which Anna here is leading. which was involved in comparing different long-read platforms. So to give you a sense of the kind of size that we're talking about, there is over 2,500 RNA sequencing-related data sets that have been released by ENCODE so far. And as I mentioned, I'm only going to focus on one section of it, which is the human long-read data sets, and I'll tell you more about it. But why long reads? I'm sure Hagen already convinced you that long reads is the only way to go yesterday. But just to repeat the obvious, if we're actually interested in understanding how it is that the proteome is diversified, if you guys recall, before the sequencing of the human genome, we thought there were 100,000 genes. And we were shocked to find out that there were only 20,000 protein coding genes, right? The result was that actually there were many isoforms that you could detect using those 2D protein gels that were coming from different isoforms of the same gene. However, it's really difficult to actually study that using Illumina reads alone. Whereas if you had truly long reads that went from the beginning to the end of your isoforms, then you've taken a really difficult problem of trying to reconstruct things and convert it into a trivial problem, which is just simply counting the full-length reads. Now, the project started in 2017. And at that point, we decided to go with PacBio sequencing. And I think that was actually a good choice. I'll admit that I now have an Oxford Nanopore GridION in my lab. But I don't think that I could be telling you what I'm going to tell you about our reads using nanopore sequencing from, say, 3-4 years ago. And let me try to show you what I mean by that. So it's worth thinking that not all long reads are equivalent. This is actually the one figure I'll show from the lrGas preprint, which is showing in the swarm plot the length of the different reads from the different data sets that were generated. And you can see that here we're plotting the length in KB of these reads on to the genome. And you can see, for example, that the cDNA PacBio is substantially different than, for example, the cDNA ONT, right? The cDNA ONT reads are longer reads than what you get with Illumina, let's say. But obviously, they're not as long as what you can get for the same material using PacBio. Now, there's another possibility, which is that it's not just about the platform, but it's also about the way you prepare the cDNA. So in fact, I would say that you can actually move this so that your mean length is not 800 base pairs, but closer to a couple KB, by just changing the underlying cDNA preparation steps. But for our purposes, this is what the ENCODE PacBio data looks like. And the reason why this is an interesting set of lengths is because the average mRNA in the human genome is somewhere around 2 KB long, which means that for many, many of the genes, we're going to be able to actually sequence from the beginning to the end of the mRNA. OK. Now, there are 160 some data sets, I believe, that are now publicly available on the ENCODE portal. About 100 of them are human, 99, I think, and the rest are mouse. They span a whole set of tissues and cell lines. And altogether, they account for at least 1 billion CCS PacBio reads. So these are the error-corrected PacBio reads. And we estimate that about 200 million of them are actually really full length. Okay, and that makes it one of the largest and most comprehensive collections of human and mouse long-read RNA-seq data sets. Okay, so that's for like the raw data part of it. Now the question is, what do you do with this data? And with long reads, we've had this ongoing issue of trying to figure out whether two transcripts are the same or not. That's not an issue when you see a novel exon or something, but it's an issue when you see that the 5' and the 3' ends are different. And we have a secondary issue, which is that we see a lot of reads that look like they're degraded reads. But we know that there are also these shorter isoforms. And the question is, can we really tell them apart or not? And I've been showing this slide for many, many years. And really, it's only in the last couple of months that I've convinced myself that we can actually take care of this problem now. OK, so when talking about long reads, we actually use these isoform novelty categories, which Anna developed in her S-Quanti paper, where we actually describe whether the isoforms that we see are matching at the level of these intron chains, known isoforms or not. When we have transcripts that match part of isoforms, we tend to call them incomplete splice matches. And the one thing that we've changed recently is that now we can look at all of our reads, and we think we can rescue about 20% of these and say that they are likely to be true transcripts. But that means that there's 80% of these that are really just parts of full transcripts, which you could try to assemble into full transcripts. But in our case, we will just let them go by the side. All right, so there are other novelty categories that you might see. For example, you might see new combination of existing exons. We'll call that novel in catalog. And then there's these transcripts that have new exons. And so those are called novel not in catalog. The pipeline that we're using was developed in my lab over the years. If I started it today, maybe I would not have just so many different steps here. Maybe I'd skip a couple. But right this moment, it starts by taking the CCS reads, mapping them with Minimap, correcting them with transcript clean, and throwing out all of the novel non-canonical splice junctions. And then we actually then look at which transcripts match which isoforms using Talon. We correct the ends with LAPA, and I'll tell you about that in a minute. And the latest and greatest is that we have now an additional package called Cerberus that we use in order to try to categorize the transcript isoform novelty. And then we have packages for doing downstream analysis, such as SWAN, which we already published. Very briefly, the way LAPA works is that we're just simply essentially going to do peak calling on ends of these reads. The key assumption is that RNA degradation is going to be less reproducible than true ends. There are still some specific artifacts, such as internal priming, that we have to deal with, but that's easily dealable. And then the very first version of LAPA that we wrote was trying to fix 3' ends. And it's really only over the last few months, as part of the ENCODE RNA working group work, that we think that we now have the TSS ends fixed up. OK, so LAPA is actually going to generate a GTF file that's got all of the transcripts that we have seen across those 160 long-read data sets. And that's a huge mess. The one thing that we decided to do was to try to see whether we could characterize our isoforms based on how different they were, both in their intron chain and their ends. And so that's actually what Cerberus is doing. So Cerberus deals with the fact that for every isoform, we can say what end it has, the 5' end, the TSS. We can characterize which intronic chain it has, and also which 3' end it has. And so essentially, it's rather than having, say, for example, I don't know, PAC6-106 for the ISO for the 6 isoform of PAC6, we can instead say, OK, well, this isoform has the first end. It has maybe the second intron chain, and it has the second TES. This allows us to now quickly start comparing isoforms by saying, OK, do they share the same 5' end? Do they share the same intron chain? And so on. As part of developing this and talking to the folks at GENCODE, I should say that I'm on the advisory board of GENCODE, we decided to always make sure that the 1-1-1 triplet, so the transcript isoform that gets the 1-1-1 designation, is going to match the NCBI ensemble MANE Select, which is supposed to be the representative transcript. If you live in that world where you don't want to think of alternate splicing, GENCODE and NCBI want to give you one transcript to represent as the transcript, right? And that's the MANE Select. So that's 1, 1, 1. And so whenever in our triplets you see that you don't see the number 1, by definition, it cannot be the main isoform that GENCODE and NCBI report. All right, so why did we break this up into 3 pieces? The goal was to try to characterize the different ways that this isoform diversity could arise. Specifically, the selection of the TSS is really about promoter choice and transcriptional regulation, more so than anything else. Whereas the selection of which exons get spliced in and out is really about splicing factors driving this. And there's a separate process that controls which polydylation site is selected at the 3' end, right? So this nicely breaks this out into 3 different components of that transcript so that we can sum up these transcripts if necessary and compare them and ask questions. Well, okay, so if we see a lot of diversity in a gene, is it at the 5' end? Is it at the 3' end or whatnot? So downstream of Cerberus, we're using packages such as SWAN to do this analysis. So I thought I would show you SWAN for the SWAN plot output for one transcription factor, which is MEF2C. Now, this is MEF2C, and I'm showing you the major isoform plotted as a percentage here across a set of cell lines. And the first thing that should stand out is that actually, depending on which cell line you're in, there's a different major isoform. Okay. The other thing that you should notice and here is that actually one of these major isoforms is missing a good chunk of the 3' end, which turns out to have the transactivation domain. And so certainly this particular isoform would be predicted to do something very different from the other isoforms that reach the typical 3' end. The third thing is that with this triplet annotation, it's pretty clear that all of these isoforms share the same TSS. It's number 1. So the annotated TSS in the MANE Select is actually the TSS that's used here. But now we can, based on looking at the other triplet numbers, see which ones share the same intron chains. I'm sorry that it's cutting off on the side here. So the purple means they're sharing the same intron chain. And these guys that have the orange arrows are sharing the same 3' end, okay, the same TES. Okay. So for me, at least, this really solves the problem of very quickly seeing where some of these differences are. Now, returning to the full data set that we have, we are detecting 65% of the genes that would be predicted to be detected based on the fact that they should have a poly-A gene, poly-A polydylation. But this actually really differs based on actually what the underlying biotype is. So we detect something like 93.7% of protein coding genes. So for most protein coding genes, we detect them. For about half of the protein coding genes, we detect them at the level of 100 TPM in at least one sample, which gives us good confidence that actually for those, we are going to see multiple isoforms and be able to reliably say what the major isoform is. This number drops down for lncRNAs, but we're still detecting most of lncRNAs. And actually, we're not detecting pseudogenes. Maybe we're detecting too many pseudogenes, I don't know. But some of these pseudogenes are obviously transcribed. So that's there. OK. Now, we're detecting something like 200,000 transcripts, of which 60% are known transcripts. And these other categories, we have 12% are rescues, 21% which are not in catalogs, and 6% that seem to have a new exon altogether. Now, how many isoforms are we detecting per gene? There's lots of ways to look at that. Here's one. I guess you guys maybe don't spend a lot of time thinking about how many isoforms there are for particular genes. But for example, there's one gene, MAPK10, that has 192 isoforms in GENCODE. And we detect about 97 isoforms in our data set. By contrast, there's one gene, elastin, which has 35 isoforms in GENCODE, but we detect 286 in ENCODE. In fact, we detect something like 250 of them in one cell line, which is chondrocytes. In that particular cell line, it's expressed as something like 10,000 TPMs, and it's just making a huge number of isoforms. It's beautiful. But there are others where we detect a lot less. All right, so that plot to me is not very meaningful. But I had a discussion one day with Lior, who's also in the audience. And when I told him about my triplet idea, he suggested, well, why don't you try to plot it as a simplex? And so the idea there is that we can essentially convert these numbers that we have here for the different genes into a coordinate where you can add each of the 3 coordinates and add up to 1. And if you can do that, then you can plot it inside of this triangle. Now, if you have a simple gene where there's just one isoform, that is going to end up dead in the center of this particular triangle. But the more you go towards one of the 3 corners, the more you're going to be doing one form of splicing. And so the forms here are going to be that if you go in this blue corner and towards here, you're doing more 5' end selection. If you're going more towards this corner, you're doing more 3' end alternative splicing. And here it would be more internal splicing. And so now we can look at these different genes, each gene across each data set, and see where they go here. Well, it's a little washed out from my angle, but I hope it looks OK for you guys. And so, for example, for that gene elastin that I was telling you about, you can see actually that a lot of these samples are really doing internal splicing. So the ends are actually staying the same. It's just the combination of exons that you're using that are changing. Whereas MEF2C is actually either near the center or it's going towards the TES. And if you remember, we are always using the same TSS for that particular gene. So the actual ratio that you're going to have is going to be something about internal splicing versus 3' end choice. And we can go look at this for any gene. There are genes where we can see actually a lot of isoforms that we detect, but if we actually limit ourselves to 90% of the, 90% of the expressed genes, if we limit ourselves to isoforms that account for 90% of the expression, then all of a sudden their behavior changes. So for example, there's lots of minor isoforms of COL3A1 that seem to be about internal exons. But the moment that you take just the isoforms that are about accounting for 90% of the expression, you either end up dead in the middle with one isoform, or you have a little bit of difference at the 3' end. OK, so I'll tell you in a minute about what this looks like globally. But first, I can now actually answer a question that I know has been burning in all of your minds, which is, how often is that MANE Select Isoform the major isoform? OK. And the short answer is it depends on whether you're looking at it as, do we detect it at all in a sample, or do we never detect it, right? Or do we only we see a switch? And the short answer is that for TSSs, actually the MANE Select is pretty good. In fact, I don't have it on a plot. I have it on another plot if you are interested. But the differences that we see are primarily not going to be at the TSSs. But the differences that we see are going to be really when you combine all 3 of them together, the TSS, the intron chain, and the 3' end together. The proportion of them where we, for example, never detect the triplet as a major isoform, right, is going to be 32%, so 1 minus 68. And this particular plot gives you the sense that there are some genes that are where you're always using the MANE Select, and there are some genes like MEF2 where you're never using actually the MANE Select as your major isoform. OK? All right. So going back now to looking at all genes globally, where do they fall if you plotted all the genes on this simplex? Well, a lot of them fall still inside of this middle region where they're either dead in the center or they're in this mixed region. But now we have these genes that end up in these 3 corners that are labeled as splicing, TSS, and TES. This is for GENCODE. This is just taking the GENCODE annotations and doing this analysis. And this is doing it with the observed isoforms that we have. And I hope that you can see that actually the proportion of genes that we're reporting as having differences at the TSS go up, as do the proportion of genes that we label as having TES, but also the proportion of ones that we label as internal splicing. So all of them actually are increased compared to the GENCODE 340 equivalent. The last part that we can now do is I mentioned that in ENCODE, we have both human and mouse samples, right? So we can actually ask the question of how often do we see the same thing for the same gene across the two different genomes, right? When we look actually across matched tissue samples in the two species, we start seeing that there are genes, I'll start at the bottom, that seem to have similar behavior. Like for example, COL1A1 in human and mouse is really behaving the same way. But we have other genes such as CTCF that in human seem to have TSS choice as the primary thing driving their isoform diversity, whereas that is not the case in the mouse data. And if you go look at GENCODE for those two, you will see that that's also reflected by GENCODE. So there are genes, very interesting genes, that seem to have different behaviors in human and mouse at the level of splicing diversity. In fact, it's only about 40% of the genes that end up in the same sector of this triangle in both species. So in summary, I told you about this data set from ENCODE that we're in the process of analyzing it. We've developed these tools such as LAPA to correct the 5' and the 3' ends of the isoforms. And this is being done to produce this consolidated GTF of all of the long-read RNA-seq data that we've analyzed. We use SWAN to look at alternative splicing between the different isoforms for specific genes or across data sets. But really, it's Cerberus that's doing the heavy lifting for us by using this triplet adaptation I told you about to really get to what kind of diversity we're dealing with. And I told you about the fact that for the majority of the genes with more than 2 isoforms, the MANE Select is not the major isoform detected. And that there are distinct changes in this isoform diversity for the same genes in human and mouse. Now, I have a huge number of people that I need to thank. A lot of the work that I told you today was really done by a really bright graduate student, Fairleigh Reese, along with a new graduate student of mine, Hassan, who've been working together on this data. And then there's a lot of other work that I didn't have time to tell you about on the mouse side that was done by Liz and Nargis. And this was all done in collaboration with my partner in crime, Barbara Wold at Caltech, along with Brian and Diane. And also wanted to thank Lior for suggesting the simplex approach for plotting all of this. And thank you to all my funding sources. Thank you.
Come back to you. Alejandro had the first question.
Hey, thank you. Great talk. Just maybe I missed this. How do you know that this is not, that you're not seeing the decay, like the transitional decay of the biggest isoform, and that's why you see these intermediate isoforms that maybe are not established and won't be really processed into proteins? Maybe I missed that. And then a bit related to that is whether, have you explored RNA binding proteins? Could those also be affecting how these other isoforms are established, or like they prevail somehow?
Okay, yeah. So I think that all the RNA that we're looking at is pretty much the steady state as measured from these bulk measurements in the tissue. Now, RNA that's in the process of degrading doesn't really have a long lifetime. And so I don't know how much of it we would see, right? Now, one of the things that we're doing to convince ourselves that these are full length is we're making sure that we see both the poly and the TSL. And so if we don't see both, we don't consider that a full-length read. It doesn't go in the analysis. This doesn't mean that you couldn't have the TSs go with the degraded RNA, but it makes it less likely. And so that was why I said with LAPA, we want to see multiple ends end in the same peak region at the 5' end before we think that that's really going to be a real RNA that we want to bank on. If we only saw it a read once or twice, and at a particular spot, that would not be enough for us to call it as an isoform. Yes,
yeah, that was amazing. Now I understand why you're doing this simplex. So why does the simplex has like kind of streak structure?
The streak structure, you mean the lines on the simplex?
Yeah,
like
exactly.
Here?
No, the when you go to all genes.
Oh, for all genes. Oh, this is probably due to sorry. Here,
yeah, that one.
Right here. Yeah, this is probably due to actually what those ratios work out to be, right? But really, there's a huge amount of isoforms that are just sitting dead center here. But there's 8,000 of them sitting here. And there's relatively few of these things here. So
they are like
I don't know
lines,
if it's about
almost symmetric.
the scaling that makes it look like there's just only a few here and there.
Yeah, I'll ask you later. The more biological question, have you looked at the isoforms that change like a protein sequence? Like what's the fraction of that?
Yeah, well, you know, our friends at NHGRI keep on wanting this in 3 dimensions so that it's like a pyramid with like the protein as the 3rd dimension.
That's what happened.
Yeah, we're working on it.
Great. Okay. We're going to take one more question while Anna sets up. Mermi?
Okay.
So thank you for an excellent talk. And with respect to the simplex representation and you showing, you know, some genes are more similar to each other than others, is there a distance measure or some, you know, measure you could kind of rank genes and start thinking about how close or far they are from each other in this context?
Yeah, I would love to come up with one. I haven't thought it through enough, but you could imagine. In our case, we just simply divided this into four sections, right? But you're right. We should be able to compute some sort of delta on this simplex to say this. And particularly, it might be useful for the human mouse comparisons to really quantify that. So thank you for that. Okay, thanks.
Let's thank Ale one more time.